Oh my Lord, it goes all the way down. Russian goes all the way to
Okay, good morning, everybody.
Yeah.
You have your quizzes back. The key is up on Moodle. Feel free to look it over and see where you missed points. Make sure that you understand where I didn't give you credit and what I was looking for. A reminder about the convo that is a week from tomorrow. Convocation will be by Rob Dunn, a biologist. He'll be talking about the role of natural sciences in understanding and dealing with global and national problems. So he's one of the movers and shakers in the citizen science movement in the US. He's written some books, he's written some influential articles. He's speaking at Convo, and there will be extra credit on the next A&P exam if you attend. So you need to attend and bring a notebook or something with you to write a 1 to 2 page outline of what he says. And then turn that in and you will get extra credit on the next exam. All right, so today we're going to pick up where we left off with glomerular filtration, and then talk about how the renal tubules, the proximal tubule, the loop of Henle, the distal tubule, and the collecting tubule, how those tubules process solute to reabsorb. Remember, we filter a huge amount of fluid and solutes, and then our kidneys reabsorb, reclaim, recover the solutes we need: water, sodium, potassium, glucose. Potassium is actually a complicated story that we'll get into on Thursday. So what we're going to do is talk a little bit more about the control of filtration, and then move on to the tubules and how they absorb sodium and glucose today, and then we'll talk about bicarbonate and potassium on Thursday. So a reminder, this is a cartoon of the macula densa and its contact with the afferent and efferent arteriole. You may have noticed on the cartoon on the quiz that this structure was not shown. The distal tubule did not go back and make contact with the afferent and efferent arteriole in the cartoon. So if you circled that area and said that's the macula densa, I didn't give you credit because the macula densa wasn't there. The macula densa is in the distal tubule, so it wasn't shown. If you said something like, this is the juxtaglomerular apparatus and the distal tubule should be here, then I gave you credit for that point. So, afferent arteriole, glomerular capillaries, Bowman's capsule, Bowman's space. Up there is the beginning of the proximal tubule, efferent arteriole, and then we'll come back. This is the distal tubule. After the proximal and after the loop of Henle, the distal tubule comes back and makes this contact. So we're going to talk about how this controls glomerular filtration. That's what we're doing right now. And the simplest way, not maybe even the most effective way, but the simplest way to understand is to notice that there are nerve fibers from the sympathetic nervous system that innervate the juxtaglomerular cells and the smooth muscle of the afferent arteriole. Nerve fibers innervating smooth muscle. So, sympathetic nervous system, the neurotransmitter is norepinephrine. Norepinephrine on these afferent arterioles the afferent arteriolar smooth muscle activate a GQ coupled adrenoreceptor, an alpha adrenergic receptor. Now remember, I could ask you again right now, I could ask you again to give me the story of how GQ leads to contraction of arteriolar smooth muscle. I did that on the last exam, but I could do it again, because not all of you got it all right. And remember that when I want the players, I want all the players. I want the details. I want the receptor, I want the G protein, I want whatever effectors, in this case phospholipase C, I want PIP2 as the substrate for IP3, I want IP3 as the product of PLC rather. I want the IP3 receptor that opens a calcium channel, so I want calcium, right? I also want GTP, because GTP is what activated the alpha subunit in order to activate phospholipase C in the first place. Remember, calcium in this story is activating calmodulin. I want myosin light chain kinase. Myosin, I'm just telling you the players, you should be able to fill in the story on your own. Myosin, phosphorylation by myosin light chain kinase, cross-bridging with actin. I could ask for the cross-bridge cycle again. In the case of the exam, I didn't ask for the cross-bridge cycle, I just asked for the names of the players. ATP, because ATP is required for cross-bridge cycling in smooth muscle, just like in cardiac muscle. So I think I just gave you the answer to that question, but it's on Moodle, and I could do it to you again. Constriction of smooth muscle. What does constriction of smooth muscle do to glomerular filtration? Oh, it says it right there. It decreases GFR by constricting blood flow into the glomerulus, lowering the pressure in the glomerular capillaries, decreasing the filtration pressure, decreasing the hydrostatic pressure would decrease GFR. So the sympathetic nervous system responds to various stimuli to constrict the afferent arteriole and decrease GFR. And some of those stimuli are dehydration, right? If your hypothalamus starts to detect that your solute concentrations are high, suggesting your water content is low by dehydration, the hypothalamus can detect solute concentrations and tell the sympathetic nervous system, hey, we're dehydrated. The last thing we want to do right now is make a lot of urine, constrict the afferent arteriole, and reduce filtration. We need to conserve water. The first way to conserve water is not filter it. We'll talk about recovering water over the course of the next hour, but one of the ways we can conserve water is to not filter. So the sympathetic nervous system detects dehydration. It also detects stress. So when you're, if in a high stress environment, you want to divert blood away from the kidneys towards muscle and brain in order to survive, then constriction of the afferent arteriole will reduce renal blood flow, which will divert blood. That large volume of blood can go somewhere else, brain, muscle, liver, etc. Questions about the sympathetic nervous system and how it operates here on the kidney?
So the sympathetic nervous system only acts on the afferent arterioles?
It primarily acts on the afferent arteriole. It probably also constricts the efferent arteriole, which would, by itself, would increase filtration because it would build up pressure here. But in combination with afferent arteriolar constriction of the efferent arteriole would aid in reducing total blood flow to all of those glomeruli and would aid in reducing renal blood flow, and that would divert blood to other parts of the body. So this is illustrating just the afferent arteriole and the effect on GFR, but constriction of both afferent and efferent would reduce blood flow overall. Other questions? Reduce blood flow to the kidney is what I meant to say. Okay, so but that one's pretty easy. There's another system that I need to introduce here that is complicated. And it's complicated because it's endocrine. It's not neuronal. So by definition, things in the endocrine system are, well, they're complicated. This one is particularly complicated. And we covered this one in the other A&P class because it's endocrine. But now I need you to apply, some of you have had this, and so you apply your knowledge from A&P I, and even if you haven't had A&P I, I'm going to give you the bare bones story here of this endocrine control. So the macula densa, remember we've got this structure, the macula densa here, and the juxtaglomerular cells. The macula densa can detect flow. And if it detects low flow, it triggers the release of a hormone called renin from the juxtaglomerular cells into the bloodstream. So, macula densa detects low flow. There is some paracrine signaling that isn't perfectly known, how the macula densa communicates to the juxtaglomerular cells. It's possible that it's a prostaglandin. That's not information that you need to know, but it might be useful to know someday, not for this course. Macula densa communicates with juxtaglomerular cells, maybe prostaglandin. Juxtaglomerular cells release renin into the blood. That's a hormone. So now we need to talk about the renin, angiotensin, aldosterone endocrine system. So renin is a hormone that is released into the blood, but unlike most hormones that interact with a receptor, renin is an enzyme. So it's a hormone because it's circulating in the body, and it's going to do all kinds of signaling. But it's not interacting with receptors. It is a hormone. I mean, it is a hormone and a enzyme. It cuts a protein that is circulating in the blood called angiotensinogen. Now, I'm introducing a concept here. If you see something that has the O-gen at the end of it, angiotensinogen, it means it's a precursor. And we're going to see that again in the digestive system. So angiotensinogen is a precursor to angiotensin. Usually the nomenclature is somewhat helpful. So renin cuts angiotensinogen to angiotensin, angiotensin 1. We're going to have two forms of angiotensin. The angiotensinogen came from liver. So when we get to the digestive tract, we talk about the important roles of liver. Production of angiotensinogen is going to be one of the list of important roles of the liver. So we'll put the liver aside for now, because that's digestive, but that's where it's from. So now the renin has cut angiotensinogen to angiotensin 1, which is a hormone in its own right. But the most important thing is that an enzyme called angiotensin converting enzyme, ACE, angiotensin converting enzyme, converts angiotensin 1 to angiotensin 2. It's another protease cut. So renin cuts angiotensinogen, and ACE cuts angiotensin 1 to the final product of angiotensin 2. It turns out that ACE is primarily released into the blood in the pulmonary capillaries. So I didn't tell you that about the pulmonary circulation, but there's another bit of information. The pulmonary capillaries are the primary source of angiotensin converting enzyme. So this is an amazing story, right? We've got the kidney, the liver, the lungs, all working together to make angiotensin 2. Angiotensin 2 is the hormone that binds to a receptor, at least in this story. Angiotensin 2 binds the angiotensin 1R, so that's the angiotensin 1 receptor. Now don't get, this one and this one are not the same one. The angiotensin 1 receptor is activated by angiotensin 2. I didn't name it. The angiotensin 1 receptor is activated by angiotensin 2, and it activates a GQ coupled receptor. So now your brain is off and running with the whole GQ story, just like it was with norepinephrine and the alpha adrenergic receptor. Now we have an angiotensin 2 activated angiotensin 1R, which is also GQ coupled. Okay, then the whole story that I gave you the players of a few minutes ago, constriction, this hormone constricts arterioles all over the place. And so by constricting arterioles in the muscle, let's see, let's go back to cardiovascular. If we constrict arterioles all over the body, what is that going to do to blood pressure? Raise it by increasing systemic vascular resistance. Remember, arterioles are the place where resistance is controlled, and by increasing resistance, we increase pressure for a given cardiac output. So angiotensin 2 is a vasoconstrictor all over the body, and it increases arterial pressure. By itself, that would tend to increase GFR, right? If we increase pressure in the arteries, then we would increase GFR. But angiotensin 1 receptors, which are binding angiotensin 2, the AT1R binds angiotensin 2, GQ coupled receptor, constriction of the afferent arteriole, and this is part of the increase in vascular resistance, and that by itself would decrease GFR. And this is typically what happens. So now we've got a complication. Sometimes, well, most of the time, what we do, what happens with activation of renin and angiotensinogen cutting and angiotensin, or ACE cutting of angiotensin 1 to angiotensin 2, and activation of the AT1R all over the body, is a decrease in GFR, because this effect overrides this effect. We still get this. We always get an increase in mean arterial pressure, but we often get an increase in mean arterial pressure and a decrease in GFR. Despite this, is basically what it comes down to. Questions? So it's worthwhile for the next exam to learn this pathway, because it is an important pathway for the control of GFR. Another pathway which I'm going to just sort of mention because I should, but we're not going to get into the molecular mechanisms of it, is another hormone called atrial natriuretic peptide. Let's parse the name out. Where is it from? The heart. So in addition to pumping blood, your heart is, the atria of your heart is actually an endocrine organ. I don't teach it in endocrinology, in the other A&P class, but you are now experts with the heart, so you deserve to know that the cardiac atria release a hormone called atrial natriuretic peptide, A&P. Okay, so it's from the atria of the heart. What does natriuretic mean? Okay, I'll give you a hint. What ion are we thinking of? Sodium. Uretic. What is uretic mean? What is it referring to? Urine. So this is a peptide hormone that increases sodium excretion. It does it by a number of mechanisms, but what you need to know is that A&P, atrial natriuretic peptide, is going to increase GFR as part of its mechanism for increasing sodium excretion. So let's go through it. Say you are retaining fluid. Your blood volume is up. Say diabetes, kidney disease. If your blood volume is up, what is detected is an increased stretch of the atria during atrial refilling during diastole. It's interesting. A stretch receptor in the atria leads to release of atrial natriuretic peptide. So it's a volume stretch detector that releases A&P that causes dilation of the afferent arteriole and an increase in GFR. And we'll talk about how sodium gets excreted in a few minutes. Okay. There's questions about these. These are the ones I want you to know. So sympathetic nervous system, number one, where renin comes from and what stimulates renin release from the juxtaglomerular cells, and the story of the RAA, the renin-angiotensin-aldosterone axis. We haven't talked about aldosterone yet. We'll talk about that later. Yes?
So for number 3, it's activated by stress. So my question is, for example, not every time we don't always experience stress when we're running.
Right.
We experience it at rest. What is the backfire to that? Because the blood is not necessarily going to our muscles because we're stagnant. But just a person under stress, would this response?
Well, remember, when you talk about stress, I think you're mostly thinking about the sympathetic nervous system. So the renin-angiotensin-aldosterone axis is not primarily activated by stress. It's primarily activated by this flow in the distal tubule, which is not really a stress response. I don't think I answered your question, but
It's okay.
okay. I prepared another slide, and I'm going to skip it. Okay? So I'm going to skip this one. This is, just so you know, there is something called tubuloglomerular feedback, which is sort of an auto-regulation. The kidney controls itself. And this is the story of auto-regulation of the kidney. Automatic regulation. It doesn't require any other input. It's not hormonal per se. It's all local in the kidney. But let's skip it. Yes?
So do we have to learn that for ourselves later?
No,
or you just,
no, we're going to skip it. This is not required for the exam. I want you to have said to the video and to you that there is such a thing as tubuloglomerular feedback and that it is an auto-regulation, but let's not get into the weeds. Questions about, so now we're going to transition out of glomerular feedback, glomerular filtration and its regulation. And we're going to transition to how the tubules work. The proximal tubule, the loop of Henle, the distal tubule, and the collecting tubule. That's the rest of this section of the course. Understanding all those parts of the kidney. Questions about glomerular filtration? Okay, so let's just talk, here's a slide from your textbook. I talked about how much water you filter per minute, right? 100 mils per minute. If you multiply that out, that's 180 liters per day. You excrete about 1.8 liters per day, and so that means you're recovering 99% of your filtered water. You're going to do in your problem set, you're going to do some calculations of excretion or filtration and excretion of sodium, filtration, and I don't think I have glucose on there, but I think I have creatinine as well. So you're going to be calculating some things related to clearance, filtered load, excreted load, comparing them for the problem set. The main point of this is that we recover 99% of the filtered water. We recover 99.5% of the filtered sodium. We recover 100% of the filtered glucose, unless we are in a situation of diabetes. And let's not worry about urea yet, but we recover almost half of the urea that's filtered. Okay, so we now have to talk about the mechanisms by which we reabsorb these key solutes. We also reabsorb potassium, we reabsorb bicarbonate. We'll talk about those later. Brittany, you're furrowing your brow. What's bothering you?
The amount, I don't know, unless I'm getting my math wrong, the urea, like, percentage looks wrong. Like, shouldn't that be above 50%?
So let's see, 24 divided by 54 is the amount.
Oh, okay, sorry, yeah, I was doing it the other way.
This is,
okay.
yeah, this is the amount reabsorbed.
Okay.
So the amount reabsorbed is 30 minus 54 minus 30.
Right, okay.
Does that help?
Yes,
Okay.
yes it does.
Okay, so now we're talking about the tubules. We're talking about, right, you understand filtration. Water got filtered, sodium got filtered, glucose got filtered, potassium bicarbonate. All those things that we need got filtered, and now they're getting into the proximal tubule. They're getting processed by the proximal tubule, the descending loop of Henle, the ascending loop of Henle, the distal tubule, and the collecting tubule. All of those do important processes. None of them are just tubes. They all do important processing of the fluid that's in them in order to recover those solids. So let's start taking it apart. The tubules are an epithelium. They are lined with epithelial cells. So here's a cartoon of an epithelium. Let's just get oriented. So this tube is lined by 1, 2, 3, 4, 5, 6, 7, 8, in this cartoon, 8 epithelial cells are surrounding this, creating this tube. The epithelium is creating the tube. And so here it is here is the tube with epithelial cells lining this long tube. So this is the tube, this is the cross-section of the tube. What are all these blobs and arrows representing? Transporters. We are going to be talking about some, not all, of the important transporters that are important for recovering solute and water from these tubules. So here's a cross-section. Here is this is a cell, an epithelial cell. And you'll see that it has it actually has 3 critical components of its membrane. And I want you to know the names of these parts of the membrane. This membrane that is lining the lumen of the tubule is called the apical membrane. The apical membrane is the one that is facing the fluid in the lumen of the tubule. The other membrane is called the basolateral membrane. Basolateral membrane. And it has 2 separate parts. The lateral part, which has junctions of one cell to its neighbor. So the lateral part of the basolateral membrane has junctions with its neighbors. The basal part of the basolateral membrane is in contact with the space, the peritubular space outside of it. And it is where there are a lot of transporters expressed. So the transporters are expressed apical and or basolateral. Apical expression of transporters, basolateral expression of transporters, and the kidney cells control not only the level of expression of these transporters, but where they go. It's an interesting area of research. How do the kidney cells know to put transporter X in the apical, or know to put transporter X in the basolateral membrane? Joey?
Can you repeat what you said about the lateral?
Yeah, the lateral parts. So this is a lateral membrane of this cell. The lateral part expresses transporters, but its main job, at least for us, is to think about its expression of these junctions with the neighboring cell. And these junctions can either be tight or leaky. So we have tight junctions and we have leaky junctions. We're actually going to need to know that information. Some of these junctions are tight, that is to say they're waterproof, watertight, and some of them are leaky. They allow things to go through. So here is the image of the kidney, or of the tubule, rather, in cross-section. Here is an image in cross-section showing the cross-section of the tubule and the space, the fluid-filled space surrounding the tubule, between the tubule and the capillary. Remember, all of this stuff is surrounded by capillaries as well. The peritubular capillary, or if we're in the loop of Henle, the vasa recta capillaries. Tubule, epithelial cells, apical and basolateral membrane, transporter expression, either apical or basolateral, junctions between the cells, capillaries that passively pick up. So now, what's the orientation here? This is another cross-section, zoomed in. So here is the apical membrane. Here's the basolateral membrane. Here are the tight junctions between the cells in the basolateral membrane. Transporters expressed here, transporters expressed here, fluid here, not a lot of space, but some fluid here, and then diffusion into a leaky endothelial lined capillary. So the capillary is passive, except for the fact that blood is flowing through it. The active transports that we'll need to talk about are in the tubular epithelial cells. And so I've been talking about this cartoon, I've been talking about this cartoon, I've been talking about this cartoon. Here is an actual image of stained cells in the, let's see, this is in the cortex. And so there are, let's just look at one of these tubules. Here's one. And this section is lined by 1, 2, 3, 4, 5, 6, maybe 7 cells. This is science here, this is not a cartoon. What are these circles? Nuclei. Yeah, that's where the DNA is. We don't talk about DNA very much in this class. What do we call this space right here? The lumen of the tubule. What do we call this space out here? Peritubular space. Maybe that's a capillary right there with catching a red blood cell in cross-section, hard to say. So you can see the peritubular spaces are tight, they're small, but they're there. Lumen, epithelial cell. What's the name of this membrane? Apical membrane. What's the name of this membrane? Basolateral. Why in the cartoon do we see that the apical membrane has all these fingers and the basolateral membrane does not? Well, that's just a cartoon of the fact that there are microvilli on the apical membrane, which is hard to see, but this is a cartoon of the way they actually look in electron micrograph. You would see this fingery projections of the apical membrane in many sections of the kidney. Why do they have that? Surface area. Increasing the surface area of the apical membrane so that it can express all of those transporters. Brittany, do you have a question?
Are we calling like the space between the vasa recta and the lubricant also peritubular space, or is there a difference between?
Let's call it peritubular space. Peri means around the tubule. The tubule, the descending and ascending limbs of the lubricant are tubules, so it's peritubular, even though the capillary is slightly different. Questions about this geometry? We need to understand this in order to understand what we're going to do with it. So now let's talk about sodium. And then we'll talk about water. And then we'll talk about glucose. I think that's the order of operation. So here's the glomerulus. Here's the proximal tubule. Here's the descending limb of the loop of Henle, the ascending limb of the loop of Henle, the transition from the loop of Henle to the distal tubule. What do we call this structure here? The macula densa of the distal tubule, or the juxtaglomerular apparatus overall. One of these is the afferent arteriole, the other of these is the efferent arteriole. Can't tell which is which. Well, I think this is probably the afferent because the macula densa is making more of a contact with it than it is with the efferent. That's a cartoonist's right. Distal tubule, cortical collecting tubule, medullary collecting tubule, urine. I didn't give you credit for urine on the quiz, but that is where we're going. Okay, so there's the cartoon of a tubule, remembering that each of these tubules, each of these segments in cross section looks something like this. So now, what I want you to remember, what I want you to know, is that the proximal tubule actively pumps sodium to reabsorb it, and water, and it's leaky. So where sodium goes, water goes because of osmosis. So the proximal tubule is a leaky epithelium. Sodium is reabsorbed and water follows sodium. The ascending limb of the loop of Henle is a tight epithelium. It has tight junctions. And sodium is going to be reabsorbed, but not water. Okay? What's the difference? Well, we're going to see many differences, but those are what I want you to know. And I do want you to know that about 2/3 of the sodium that we filter is reabsorbed in the proximal tubule. We're going to talk about mechanisms in just a minute. About a quarter of the filtered sodium that was not absorbed by the proximal tubule is absorbed by the ascending limb of the loop of Henle. Remember, we're not caring about thin or thick very much. So the ascending limb reclaims 25% of the filtered sodium. The proximal tubule reclaims about 2/3 of the filtered sodium. And then we'll talk about the distal tubule and the collecting tubule. It gets the rest. So if you add these up, I think it probably adds up to about 100%, but we don't recover 100%, we recover 99.5% of the filtered sodium. There's a round-off error.
So sodium is pumped into the tubule, or is it pumped out?
Sodium is going to get pumped out of the tubule to be reabsorbed by the blood so that it's recycled. We don't want to urinate away all our sodium. When sodium is pumped in the proximal tubule, water follows it because the epithelium is leaky. Okay, so now, here's a really hard slide. Let's do this. Gave it to you big. We'll do this and then we'll take a break. So this is a cartoon of a proximal tubule cell. Apical, basolateral, junction. Okay, so this is the lumen, and this is the blood. No, sorry, I'm getting this wrong, sorry. This is apical, scratch what I just said. This is apical. This is the apical membrane. This is basolateral over here. I don't know why they drew it with wiggles because it should be nice. It shouldn't have wiggles on it. Apical, basolateral, junction. With me? Lumen, peritubular space. It's called here interstitial space, whatever. Peritubular, apical, luminal, lumen. How do we get, let's just talk about this one first, and then we'll do some of the others. How do we get sodium that we have filtered back into the blood in the proximal tubule? Remember, 2/3 of it is going to be recovered by this section of the kidney. Okay? Let's do that. So the first thing to know, and I'm going to circle it because I do want you to know it. What is that? That's the sodium potassium ATPase. You've seen it before in this class, you've seen it before in CalPhys. It's going to be really important. Which way does it pump sodium? So it's a primary active transporter. Which way does the sodium potassium ATPase pump sodium? Out. From the cytoplasm into the peritubular space. That is one of the important ways that we get sodium across this membrane. Now let's talk about how we get sodium across this membrane. Right, they have two membranes in parallel. We've got to get it across the apical membrane and across the basolateral. There are two main transporters for sodium across this membrane. The sodium glucose co-transporter and a sodium proton exchanger. So sodium is pumped out of the basolateral and it flows down its concentration gradient apical. One of these transporters, they're both secondary active transporters, but it's passive flow of sodium. Sodium is going down its concentration gradient into the cytoplasm and then pumped out the basolateral. So now we have, right there, we have the path, at least most of the path, for sodium recovery. Is it clear how sodium is moving from here to here, which is not shown, to here? We are pumping it across this epithelium. So what else do we need to know? We also need to know that there is a sodium bicarbonate co-transporter on the basolateral membrane. So bicarbonate is going to help pump sodium out this basolateral membrane. And where does the bicarbonate come from? And where does this proton come from? It comes from CO2 and carbonic anhydrase. We have carbonic anhydrase in all of these cells. Where did the CO2 come from? Well, the CO2 diffused from the blood and from the urine into the cell. CO2 goes wherever it wants to go. Remember, it just diffuses. We have 40 millimeters of mercury of CO2 in the blood. So we have 40 millimeters of mercury in the peritubular space. So we've got plenty of CO2 inside the cell, and it's being converted by carbonic anhydrase into bicarbonate, which is co-transported with sodium basolateral, and protons, which is exchanged sodium apical. Those are the pieces of sodium transport in the proximal tubule that I want you to understand.
What about the one with the water?
Okay, what about that? So I can't reach the one up there that shows the sodium going backwards, cross out. You don't need to know that. The one up there, this one here, is actually referring to glucose. We'll come back to that later. It's not sodium. Remember, we're in the proximal tubule and the epithelium is leaky. So water, if sodium is being pumped, water is flowing to accompany the sodium. And the water is flowing in this paracellular. So this is called transcellular pumping of sodium, and this is paracellular flow of water. So I do want you to know that there is paracellular flow of water because of the leaky epithelium. But let's not worry about this paracellular sodium movement. So we don't have to worry. It's true, it happens, but let's not worry about it.
All the other mechanisms are transcellular?
Yes. So let's take a break now, and then we'll come back to some other sections of the tubule and answer your questions. Let's take a break.